Okay, so as promised, on page 320, they have this really great picture of how HIV is basically transmitted into the cell and how replication happens. And to me, kind of the, if you will, the bread and butter of HIV and how things happen inside of the body. So I thought it was really important to do some sort of a kind of a mini lecture on this process because in my opinion, explaining it all in one process is going to help in understanding it. So I have this as obviously the background. So we're just going to kind of take a look here. And just as a side note, originally I was like, okay, I'm going to have a list of medications for them to know this and that and whatnot. I really want to keep this basic because I want you to understand it and know there's a medication for it. I want to be there right now. So that is where we are. Okay. So what we're going to do is we're going to start with the beginning part, which we talked about a little bit in our first lecture, but, and obviously we can all read, but I kind of wrote it as shorthand and kind of went over it and making it a little bit of an easier, hopefully kind of shortening the words. We don't need to know every single word and all. I want you to get the big picture of what's happening. So basically the first, remember there's there's a 7-part transcription process. Okay, you might want to know that or keep that in mind. That may or may not be on your quiz on Friday. So number 1, that is having to do with the attachment part. So that phase is basically where the HIV is somehow gotten into the body through, either HIV, or I'm sorry, injecting oneself with a used needle. It could be from a sexual behavior. However, it gets into the body and basically attaches itself to a CD4 cell. And a CD4 cell is what? A CD4 cell is a white blood cell. It's a lymphocyte, same thing. And in essence, it is a T cell. So now we have this foreign HIV pathogen in our body that we don't want. And the takeaway from that is there's now a medication that is called, we're not listing the medications right now. We want an overview because I feel like this is such a cumbersome topic. So in order to stop the binding from happening, they're giving medications that are called CCRS antagonists. So just stick with me here and kind of just engulf this part of it first by understanding that there's a medication to stop this replication. So what that would mean is as the HIV virus continues to be in the body, because we know it doesn't go away, this medication, when a patient is prescribed this medication, and as I'd mentioned in earlier lectures, that we're putting medications together. So instead of taking, 20 a day, you may take anywhere from 2 to 10 a day. It just depends on the person, what they're taking, why they're taking it, if they have another maybe infection or whatever may be going on. But now we have this medication that can stop this from happening, which means it's going to lessen the viral load in the body, sometimes to potentially an undetectable amount, right? So this is a very, very, very good thing. It is helping to where HIV is not able to bind to the CD4 cell, which is the T cell. And the T cell, remember, that's like that really important part where we can tell, do we have HIV or do we have AIDS? And what number is that? I'm not going to tell you because that is on your quiz and I'm not going to give everything away for free. So there's that part there. And then we're going to move on to the second part, which is where we have fusing of certain things happening together. So now we have HIV inside of the CD4 cell. So it's fused, the HIV is now inside of that membrane, and they're together, which means that because of this, it allows HIV to enter the CD4 cell, and we don't want that. We don't want we don't want HIV inside of the CD4 cell, right? And there, as it says here, stops fusion inhibitors. That is a medication. Is a medication which is under the classification of a fusion inhibitor that will stop this from happening. So it actually stops it just from HIV being able to go in the cell in general, which is great. So again, all of this is making it to where our body and our immune system is able to work as properly as possible. Then we go down to the reference transcription. So this part it's a little more wordy, and I don't like that, so I'm going to make it less wordy. And basically at this point in the process of HIV, so we're pretending there's no medications on board. I'm just showing you that there are now medications on board to prevent this, and this is why people are able to live so long. So I'm just showing you the cascade and the fact that now we have medications that can prevent this. But on the third part of the cascade of HIV replicating and going throughout the body and doing what it wants to do and feels it needs to do to our immune system, we have phase 3, which is the reverse transcription. Now, what does that mean? That's a lot of words on there. So I'm going to kind of shorten it for you a little bit. And basically at this point, if no medications are taken and we're not, let's pretend we don't even know we have HIV. Now we know that HIV is inside the CD4 cell. We've already talked about that. And we know that the CD4 cell is the T cell. And we know that the T cell is a white blood cell, which is also a lymphocyte. Again, knowing the terminology can be important because people use different words, different terminology. So HIV basically has an enzyme, and it's converting the HIV genetic material. Basically, HIV wants to go and go and go and go and replicate and replicate and replicate. And in order to do that, or not in order to do that, but it's doing that inside of the body. And of course, we don't want that to happen. We don't want more and more and more and more and more cells to be infected with HIV, because we know what HIV does, and that's going to then hinder the immune system, hinder the C4 cells, hinder the T cells, and then we know that that can change HIV into AIDS. And we don't want that to happen. And there are medications that can make that from not happening. And as we see here, it says stop and stop. So what stops it? These types of medications, and NRTIs as well as NRTIs. So that's great. That's going to stop that part of it. We've come so far, by the way, if you've never really gotten to the nitty gritty of all of this. Oops, my apologies. Okay. Then we're going to go to number 4. And this has to do with integration. So integration has to do with once the CD4 cell, its nucleus is imposed with the HIV release, because remember, we talked about that enzyme. So that enzyme is basically making it to where HIV can get into the nucleus of the CD4 cell. And if we remember from biology, the nucleus is a very important part of the cell. We do not want this. This is very, very dangerous, and it can cause a lot of havoc on the immune system and can cause HIV to grow at a very rapid rate. So we do not want this. So we have that HIV releases that enzyme, and then HIV uses this to insert or integrate, whichever word you prefer, it's viral DNA. And remember, DNA is everything. Your body is being taken over by HIV DNA. We don't want that. Remember that. HIV DNA. And that's not on a test. That's just, it's hopefully helping like to kind of click. You do not want that to happen, because once that happens, HIV is taking over, right? So we have DNA of HIV, and it is basically going into, I actually want to read this exact sentence. It's viral DNA into the DNA of the CD4 cell. So now we have HIV DNA going into the CD4 cell DNA. And what do you think it's going to do with that? It's not going to leave it alone. It's going to change it, mess with it, and it wins. So we have a medication for that. What's going to stop that? Integrase inhibitors. So we have a medication for that as well, which again keeps our viral load where it needs to be. Beautiful. Then we have our 5th process. Remember, we have 7. And that's where replication takes place. And replication, I think we're all familiar with that word, is making more of. So once we have the integration into the CD4 cell, and there's DNA, we have HIV DNA inside of the CD4 cell, HIV will begin to basically use its own tactics of the CD4 cell to make long chains of HIV proteins. So basically HIV now has taken over the CD4 cell, which is a T cell, which is a white blood cell, which is a lymphocyte. I said that again twice. So HIV is now in there. It has taken over the nucleus of the CD4 cell, and it's making HIV proteins. That is not a good thing. And it's making a lot of them because it wants to spread it throughout the body in order to infect the body to where the immune system is not able to fight it off. These protein chains will then build building blocks for more HIV. And that obviously we know is not good. Then we go down to the sixth part, which has assembly. And if it doesn't say stop or have any other things, there's not a specific medication to help with that. That doesn't mean all the other medications aren't helping, obviously, to avoid to get to the replication phase. So just because it doesn't have a specific medication doesn't mean that all the other medications aren't helping in general, if that makes sense, I believe. And if not, we have a discussion board, and that is why it's there. So now we're at phase 6, and that is the assembly phase. So at this point, we have HIV proteins. So we already know that things are starting to move along. We have DNA that's being made with HIV. It's being made, it's being spread. And the HIV proteins and HIV RNA, and I don't want to get too far into RNA. And I mean, we know what DNA is. RNA is very, very similar to DNA. The main difference is there's 3 main differences. I don't think it's completely important, in my opinion, for the understanding of HIV. But if you see RNA, it has 1 strand. If you see DNA, it has 2 strands. I will tell you that. That might be somewhat important somewhere down the line if you see a picture of it. So we have HIV proteins and HIV RNA that are going to be moving to the surface of the cell and assemble itself into an immune non-infectious HIV. Not non-infectious, an immature HIV. So at this point, it's not to the point where it's able to cause HIV. It's like a baby HIV, but it's going to be moving throughout the body. Okay. Sorry, I'm just trying to get to the 7th one. And again, there's no specific medication for this, but at this point, we do have HIV happening in the body. We have, it's doing its thing, it's spreading, and it has a plan, if you will. And I love this picture because I think it depicts it quite well. And you can go back and listen to this and kind of like watch as I'm going down. And I forgot to use my little pointer. I was going to do a whole thing, but I think this is good enough to get the big picture of what I'm hoping you get from this. So then we're going to come to the 7th phase, which is called budding. Now, I am not going to ask you, what is number 1 called? What's number 2 called? What's number 3 called? Up to number 7. Do I think that's important? No. Do I think it's important that you understand an idea? You don't need to know every single nitty-gritty part of how HIV happens, stops, goes, this, that, whatever. The point of this is to know that there are 7 steps. It is intricate. HIV is very smart, and there are medications to stop certain acts in order for people to be able to live longer. So if a patient were to ask you, wow, back in the day, like people were not living as long, like why? You can answer that question by saying, well, we've become much smarter in the way of understanding how HIV works in the body, what it does, and we've been able to create medications in order to stop these processes so that we can catch it, stop the process in order to not prolong whatever process that is, so that more HIV is not made and we can keep our viral load where it needs to be and people healthy. And unless someone's not aware they have HIV or not taking care of themselves the way they should or taking their medication, unfortunately, I mean, the medication is key really when it comes to HIV, as you can see. Okay, so going back to just the last part here is that, and this one I had planned on just kind of reading from. So budding has to do with the newly formed immune non-infectious HIV, which will push itself out of the host cell. Remember, we talked about the host cell. The host cell is the first cell that was infected by the CD4 cell that was first infected. That's the host, right? So that is an infected cell. And as it's doing all of its stuff, we already talked about HIV took over the nucleus, it made the DNA, it did all of its stuff. It's pushing out HIV that is baby HIV is what I call it. And now the new HIV, it's going to release certain, enzymes in the body, basically what we were talking about earlier, in order to make more. And things are going to break along with different, and I don't want to get too nitty gritty, but basically lots of different things are involved here. I mean, we have enzymes and proteins and different types of immune viruses that are going out throughout the body. But the main point of this last phase is that the smaller HIV proteins basically combine to form mature HIV virus, right? So now it's basically left the host cell, did what it wanted to the host cell. So it does pretty much almost everything within the host cell until the end, where it says, okay, I'm leaving now. I'm going to go infect more cells. I'm going to do my thing. And then at that point, it's where we're going to spread all over the body. And there is a medication for that, because it says stop. And that medication is our classification of our proteinase inhibitors. Inhibitor meaning stopping this from happening. So I know that this was quite quick. I personally found it very helpful when I was learning HIV, because it is so, and even, I mean, things have changed so much since when I learned about it. But what I find very helpful and useful is going to this page, looking at it, and writing it in your own words. Because proteinase and write it in English, like words you understand, and make your own chain. And that to me is very, very important, because then you can see it and understand it in a way that makes sense. The book talks a little bit about medications. I don't personally think the medication names are as important as knowing that there's a medication that stops it and what it does to it. How does it stop it? Does it inhibit something? Is it moving something? What is it doing? Most of them are obviously inhibitors, but this is what I personally think would be helpful. I do not plan on testing you on medications for any of the, I'm going to stop the share, sorry, because I have information in the back that I shouldn't be showing. I'm not asking about any of the medications, and you all know I love medications, not because I don't think it's important, but because I more want you to understand the disease process and that there are medications to do certain things to it. And you can read it in the book. And once you get that down, like, okay, this is happening, this is happening, this is happening, number 5 is happening. Okay, there's the 7 steps. These have medications. Okay. And then if you can remember 1 or 2 medications from it, great. If you don't, that's why you have a med book. But that was kind of my whole goal for today. So I hope that this made a little more sense in the fact that it can really be broken down to not be as difficult, because there's all these big words and it's kind of overwhelming, and I don't want it to be. So I hope this did help to some degree. Again, we have the discussion board. Please let me know if I can answer questions about it, but just wanted to give you that, and I hope it helped. Thank you.